My name is Doug Sellinger, and I'm the founder and CEO of Plex Research. Today, I'm going to show you how Plex can be used to access massive amounts of drug discovery data, and how it can make large language models like ChatGPT far more powerful for life sciences research. Large language models are trained on textual data, like scientific publications, patents, and web pages. The problem is that the vast majority of research data is not stored as simple text. The literature is filled with large-scale experimental data sets. They're complex, statistical, dependent on biological context. They're noisy, they're highly diverse, and they include high-value data sets like high-throughput screens and multi-omics data. So what if you need to know if inhibiting your target will have a therapeutic effect, or if your drug will cause a safety issue, or which patients will be most responsive to your therapy? The answers may be hidden in these large-scale data sets, and Plex can help you find them. Plex can identify where multiple sources converge on an answer and shows you the source of every data point, so you can understand the strength of the hypothesis, what caveats to consider, and what the follow-up should be. Plex can make connections that have never been made before, making it a true discovery platform. Together with a large language model like ChatGPT, we can integrate and use scientific data of almost any type. We've worked with dozens of companies, including some of the largest pharma companies in the world, and we've covered a wide range of use cases spanning multiple disease areas and therapeutic modalities, including both preclinical and clinical projects. The applications are diverse because our data is diverse. There's transcriptomic and proteomic data, from genetic modulation, from drug treatments, disease processes, and toxicities. So given the gene signature, we can identify what might have caused it. There's massive amounts of chemical structure and bioactivity data, so we can infer potential drug targets and their mechanism of action. There's a huge amount of cancer genetics data, including the Cancer Cell Line Encyclopedia and the DepMap, so we can identify the genetic drivers of cancer and match the right drugs to the right patients. It's really an incredibly flexible platform. I'm going to cover two demos next to show you how this actually works. In the first one, we're going to investigate the target and mechanism of action of a series of antimalarial compounds. They've never been characterized, so we're actually going to come up with an entirely novel target hypothesis as part of the demo. For the second one, we'll show you how we can identify the nature of a skin lesion using a list of differentially expressed genes. As you'll see, we're able to do this with a remarkable level of confidence and specificity. In both demos, we'll bring in ChatGPT to interpret and integrate the results, combining what Plex found with what it knows from its prior training data. Before I get into the demos, let me describe briefly how we work with our customers. We can provide Plex as a quarterly or an annual research service where our scientists run the analyses and present the results to you, including all the supporting data. You'll also get direct access to Plex so you can run your own analyses and follow up on what we've done. Or we can integrate your organization's proprietary data and algorithms into a private, secure instance of Plex as an enterprise-level solution. If you'd like to learn more, please visit our webpage at plexresearch.com. And now on to the demos. In this first demo, we're going to have a look at a series of antimalarial compounds that came from a phenotypic screen from GSK. And what we're going to do is paste in the SMILE strings, so the structures. This works with entirely novel structures. Here I put in the SMILE strings. They get mapped to IDs and structures here that you can see. So these are 7 compounds in this series, and we're going to run this search. As you can see, the top targets are phosphotransferases, and these are at the top of the list because there's more data supporting these targets than any others. And that data comes from chemical similarity, where we've identified a number of analogs that have similar structure to the input compounds, which are on the left here. And these compounds are phosphotransferase B inhibitors. And we can come down and see the level of similarity, both the scores, but also the structures. And we can hover over these data points and see the exact assay that was used to make that measurement. These can all be clicked on, so we can go back to the original data. In this case, it's coming from Kemble, and there's even a paper associated with it. So we can see the original paper that generated that activity data. So we can get a strong sense of the data behind this, both the chemistry, the biochemistry, the source of all these data points, how many data points there are. Here there's more than 100 connections. And so this is all data that can be evaluated. As you can see, there's a lot of data here across a lot of categories. So we're going to bring in ChatGPT to interpret the results. I'll launch the large language model integration window. We'll paste in the query and we'll submit it. The prompt starts with some background about Plex, and then it sends the Plex result to ChatGPT. And then we ask, what do you think the target of the query compounds might be? And ChatGPT looks across the different categories and comes to the conclusion that perhaps these compounds are farnesyltransferase inhibitors. And that's based on information from the target page that we looked at before, which again is coming from chemical structure. We found similar compounds that were farnesyltransferase inhibitors. It's also finding data from the publications page, which is also driven by chemical structure, because again, we put in the structures. Plex found similar compounds that were mentioned in these papers that describe farnesyltransferase inhibitors. So ChatGPT is picking up on that and adding that to the evidence that supports this farnesyltransferase hypothesis. Now, what's also interesting here is that it's clear that ChatGPT understands that farnesylation is a part of prenylation. It's mentioning that in the response, and that prenylation is an important part of the malaria life cycle. So it's actually piecing together, first of all, this idea of farnesylation being a kind of prenylation, and then linking prenylation to antimalarial activity. So we've got, when we put it all together, we're able to go all the way from just a set of chemical structures to a putative molecular target that has support from multiple sources, and that furthermore is linked to the biological phenotype of interest here, which is antimalarial activity. So we've got this full traversal from the chemical structure to molecular target to biological activity with support from multiple sources. So it's really a powerful way to bring this all together, develop well-supported hypotheses that in quite a lot of detail. In this next example, we're going to use a list of differentially expressed genes to investigate the nature of a skin lesion. And I'm going to paste into Plex here the gene list. These are just gene symbols, and Plex takes them and maps them to all the proper IDs and descriptions. And then we're going to run this as a search. And Plex is going to take that gene list and look across a huge amount of data and ask, where have we seen something like this before? Now, you can see there's lots of different data returned here. We're going to focus first on the RNA profiles category for the purpose of this demo. And what we can immediately see is that the genes that were differentially expressed in this lesion are very similar to independent gene expression studies of psoriasis patients. So all of the top results here are all gene expression profiles from psoriasis experiments. For any one of these, we can click on the results, just like in anything with Plex, and see what the underlying data is. Here we can see which genes are involved in that overlap. There's 7 altogether, some of them with multiple comparisons in that study where they were changed. And we can go through and see exactly which genes were changed and how much. So there's that transparency and that traceability back to the original data. But what we're going to do is bring in ChatGPT to look over this result and really summarize across all the categories what data is there and what kind of lesion that data supports. So I'm launching ChatGPT here. I'm going to paste in a prompt to investigate the skin lesions and paste that in and hit return. And we get the results back here, and you can see that it's concluded that the lesion is likely to be related to psoriasis. And this is coming from, first of all, the RNA profiles category that we looked at, that there are a number of studies, independent studies, where similar sets of genes have been modulated. So that's supporting this hypothesis. Furthermore, it looks at other categories like the disease category where psoriasis is ranked very highly. Also infection, it points out, which may be linked via inflammation. And then it goes to the protein complex category, finds additional supporting evidence there. And here it's actually bringing additional information because it's saying that the calprotectin heterodimer and also this S100 complex are highly ranked. And it's bringing in the information that both of these have been implicated in psoriasis. So it's adding some additional dots that it's connecting, taking what we find in Plex, which again is at least data from at least 3 categories, and in these categories, sometimes multiple data points, taking all of that, combining it with what it knows about psoriasis and various known molecular mechanisms of psoriasis, bringing that in, bringing it together, and saying, considering all these findings together, it is highly likely that the characterized lesions are indeed related to psoriasis, which in fact they are. The source of this data was in fact a psoriasis study comparing lesion skin to normal skin. And so it's getting the right characterization, a relatively detailed characterization, and there's really a lot of confidence because it's able to do that using information from all these sources. But again, the only input was the gene signature. We didn't tell it anything about psoriasis. And so it's really a very data-driven method. The particular study that was used is not here, so it's not. So we're actually doing this through matching to completely independent studies, and so in a very data-driven way, we're getting to the lesion type, and we're able to show precisely the supporting data behind it. I hope you enjoyed the demos and saw how powerful the combination of Plex and ChatGPT can be. If you'd like to learn more, please visit us at plexresearch.com. We'd be happy to set up a time to discuss our technology, answer any questions, and explore how we can help you reach your scientific and strategic goals. Thank you for your time and attention.